In this video, we'll be learning some important concepts about the science of nutrition. Basically, how do we know what we know about good and bad nutrition? How do we really know what's healthy and what's not healthy? The answer is science. To begin talking about nutrition science, we first need to understand a little bit about how science is actually done. When scientists are interested in studying a particular area, they first identify the problem they're going to study. Sometimes this can take the form of a question that they want to answer, such as, what does vitamin C do? How can we prevent cardiovascular disease? Once the problem or area of research has been identified, scientists look to gather initial preliminary data. They may gather this data by simply making observations about the world around them. Oftentimes, in the scientific realm, that data is gathered by looking through the published scientific literature, basically figuring out what other people have already done and already found. After gathering preliminary data, scientists develop a hypothesis. A hypothesis is basically a possible explanation, a possible answer to the question the scientists have asked. A good hypothesis yields specific predictions in the form of, if this hypothesis is true, then this will happen, or then if we do this, we'll observe that. These predictions can then be tested experimentally. This testing predictions following from the scientist's hypothesis, this is the research part. Once the research has been done, once you've done the experiments, you've tested your hypothesis, you ask yourself, do the new data agree with your hypothesis? If the answer is yes, then you continue testing the hypothesis. You may need to repeat the experiment, you may need to test it in slightly different ways. If the answer is no, the new data do not agree, then you may go back and figure out what other possible explanations could exist. Come up with alternative hypotheses. I want to stress that the scientific method is not just something that scientists and academic research do. The scientific method is something that we all practice every day, every time we encounter a problem or some situation that we aren't quite sure what's happening. For example, let's say you have a flashlight and you are worried that the storm might be coming and your power might go out and you want to test and make sure this flashlight works. So you turn it on and the flashlight doesn't work. Problem, flashlight doesn't work. Well, so you think about what you know about flashlights and what flashlights need in order to work, and you generate a hypothesis, such as maybe the batteries are dead. That's a hypothesis. That's a possible explanation for your problem, the flashlight not working. Now, your hypothesis that it's the batteries leads to a very testable prediction. If I replace the batteries, the flashlight should work. You test that hypothesis, you replace the batteries, you turn the flashlight on again, and you see what happened. If the flashlight turns on again, bingo! Hypothesis confirmed, and your flashlight's working now. If the flashlight still doesn't turn on, well, you need to go back to the drawing board. Maybe it wasn't the batteries. Maybe the bulb is out. Maybe something happened to the flashlight and it's not making the connection anymore. The point is that you've just followed the scientific method without even really thinking about it or even realizing it. You've identified a problem, you've generated a hypothesis and predictions from that hypothesis, you've tested your hypothesis, and you've figured out if the data agree with your original hypothesis or not. Now, there are other things you need to consider when you are conducting scientific research, and one important thing that you need to think about is what controls do you need. When you're thinking of controls, think about what else could possibly explain my observations, and that tells you what you need to control for. If you think of our flashlight problem from the previous slide, your hypothesis was that the batteries were dead. An alternate hypothesis, something else that could explain your observations, would be that the light bulb was out in your flashlight. Now, if you came up with these two hypotheses and you replaced the bulb and the battery, your flashlight might work, but would you have actually figured out what the problem was? If you replaced both the battery and the bulb, the flashlight might work now, but you don't know which was the problem. You don't know if it was that the batteries were dead or that the bulb was out. If you want to know whether it was the batteries, you need to control for the bulb. Keep the bulb in the flashlight and only change the batteries. If you're performing an intervention trial, something where you are giving a particular group of people a vitamin supplement, say, or any kind of drug, then you need to make sure that you have both a control group and you have an experimental group. The experimental group is the group that's getting the treatment, the supplement or drug. The control group is basically your comparison group. Ideally, you want your control group and your experimental group to be as identical as possible, except for the single treatment that you're studying. For example, what if I wanted to test, is vitamin C effective against colds? You treat one group with vitamin C, that would be your experimental group. You treat the control group with nothing. They don't get the vitamin C. All right, so far so good. We've got our experimental group and our control group. But now, what if we didn't do a very good job of designing our control group, and we find out that we had a bunch of 20-year-olds in our experimental group, and a bunch of 80-year-olds in our control group that didn't get vitamin C? Well, we'd probably find that the experimental group didn't get a lot of colds. However, did they not get colds because of the vitamin C? Or was it just because they were younger and healthier with better immune systems? One important type of control to think about if you are testing a new drug or a new nutritional supplement is the placebo control. A placebo is also known as a sugar pill. What it basically is a pill or some kind of treatment, or in this case, a surgery that contains everything involved in taking the drug. Looks the same as the drug, you take it as frequently as the drug, except that it doesn't have the actual drug or the actual vitamin supplement. In our vitamin C example, the pill would be the same size as the vitamin C. You'd take it as often, but it wouldn't actually have any vitamin C in it. Placebo controls are important because there's a not well understood, but well-known phenomenon in medical research called the placebo effect. The placebo effect basically is the observation that patients will report that they feel better and actually feel better, and sometimes actually even do better if they think they're getting treatment, even if they aren't. So even if they're just getting a sugar pill, no drug, no actual supplement, patients feel better. Sometimes doctors use this placebo effect to their advantage. If they have a patient coming into their office demanding some kind of drug, but the doctor doesn't have a drug that would actually help them, doctors have been known to give patients placebos just to make them feel better. There's a report not that long ago that said about 50% of doctors had prescribed placebo pills. It said the placebo effect isn't really well understood, but we do know it happens. And the really weird thing is that there was a recent report that says the placebo effect works even if a patient is told it's a placebo. Even if you tell a patient they're just getting a sugar pill, that the drug isn't actually in it, that makes some patients feel better. Okay, you have identified your problem, you've generated your hypothesis, you've done the experiments to test your hypothesis. Now you need to analyze your results. the main question that you want to ask yourself is, are your sample groups different? Are your experimental group and your control group actually different from each other? The quick way to compare two groups is to look for the mean. The mean is the average of your measurements. If the mean effect between two populations is the same, the populations probably aren't that different. Your control group and your experimental group aren't different, and the drug or supplement you are testing doesn't have an effect. If the means are different, that could mean that they are actually different, that the two groups are actually different. However, it would take a much deeper course on statistics than we're getting into in order to really fully determine if two groups are different. The mean is just a quick comparison. One thing that I do want to mention you should look for when analyzing data is the variability in a sample. And this is often given as a standard deviation. Sometimes if you are looking at reports in a newspaper or magazine, you might see a plus minus. So like 50 percentage points plus or minus 2. That would mean that the actual mean could be between 48% and 52%. I want to illustrate for you briefly the importance of considering variability in a sample. So let's say we had two groups. We had a group A and we had a group B, and we were comparing these groups on, let's say, weight. Right? Now, group A, I'm going to draw dots on this graph, and each dot represents a single individual in group A. All right? Now let me draw the dots for group B. With me? In group A, all of the dots are roughly clustered around the mean. In the case of group A, the mean does a pretty good job of giving an indicator of what that population is like. Now let's look at group B. Group B has a lot of low dots, but it also has these two high dots up here. So the mean for group B is also pretty much where the mean for group A is. If mean was the only thing you looked at, you might think that group A and group B were very similar. But when you look at the actual individual data points, these two groups are actually very different. If you didn't look at the variability in a sample, you might miss that. The second thing you ask yourself when you're analyzing your data is, do your results support your hypothesis? Scientists don't really like to say that results prove something, and that's just because scientists know that you might think something's been proven, but if someone else comes along with better data, you might find out that you were wrong. So what they say is, our results support our hypothesis, or not. Another way to think of this question is basically, did I see what I expected to see based on my original hypothesis? Now, there's basically two possible answers here, right? You could have no, and you could have yes. The actions you take after coming to these conclusions depends on whether you answered no or yes to this question. If your results don't support your hypothesis, you might ask, well, did my experiment work? And part of checking on if your experiment worked would be to maybe check your controls. Did your control group do what you expected them to do? Check controls and ask if the experiment worked. If your controls look fine and the experiment seemed to have worked, well, then you might go back to the drawing board and come up with a new hypothesis. If the answer is yes, that your results do support your hypothesis, the most important thing that needs to be done is to repeat It sounds redundant, right? Like your hypothesis is supported, your hypothesis is probably true. Why do I need to repeat this? Well, in science, scientists acknowledge that things can go wrong, and things that seem to have worked perfectly on Wednesday, you do them again on Thursday, and you might get completely different results. So scientists don't believe anything until it's been repeated a few times. My molecular biology professor used to tell me, if it worked the first time, you know you did something wrong. Perhaps even more important than repeating your experiment yourself is seeing if others can repeat it. Something that only works in one lab, in one particular population, in one particular researcher's hands, is not likely to be useful. It's not likely to apply to everyone out there that's anxiously waiting to figure out the results of this experiment. You need to see if others can get the same results, if others can generate data that supports the same hypothesis. Then you know you've got something intriguing. As you're analyzing your data and you're coming to conclusions, one really important thing to remember is that correlation is not causation. What this statement basically says is that just because two things happen at the same time, it doesn't mean that one causes the other. Even if one thing happens before the second thing, it doesn't mean that the first event causes the second event. Sometimes things are just a coincidence. Or sometimes they're related in ways that aren't directly causation. For example, my favorite example is that this really great earth-shattering observation I've made. Every night I go to bed, and every morning when I wake up, the sun is up. It's amazing. Every single night I go to bed, and every single morning the sun comes up. That's a correlation. 100%, every single time. Does my going to bed cause the sun to come up? If that's my hypothesis, it would probably be a pretty easy one to test. All I'd have to do is not go to bed one night and see if the sun still comes up. It'd be a pretty easy one to disprove. But it's a great example of how correlation does not equal causation. Here's another example. There's a great website out there called Spurious Correlations, where one Tyler Vigen has generated a computer program that goes through and analyzes data from the CDC and from the USDA that looks for data that correlate with each other. This is one example of a correlation that his program found. The green line is consumption of cheese, and the blue line is people who die by becoming tangled in their bed sheets, which I personally did not know was a thing, but the CDC apparently has data on it. If you look at these two lines, you see that as consumption of cheese rises, the number of people dying by getting tangled in their bed sheets is going up as well. Does this mean that cheese consumption causes people to get tangled in their bed sheets and die? It might seem to imply that, but remember, correlation is not causation. Here's another one that's a little more questionable, and this is an area that researchers are actually still trying to figure out, and that's the correlation between obesity and metabolic disease. Like type 2 diabetes, increased risk of cardiovascular disease. There is a lot of evidence that individuals who are obese are more likely to develop metabolic disease. And there are a lot of health programs devoted towards reducing obesity in the hopes of reducing metabolic diseases. However, as I said, this is an area of active research. And one thing that people are very interested in is that there are factors that lead to obesity that by themselves are also linked to metabolic disease. So things like leading a sedentary lifestyle, not being very active, spending a lot of time on the couch in front of the TV. Things like overeating, taking in too many calories. Things like having a high sugar intake. All of these things lead to obesity. But here's the thing. All of these factors, they also lead to metabolic disease independently of obesity. If you are thin, but you're sedentary and you overeat and you have a high sugar intake, you're more likely to develop metabolic disease even if you're not obese. So the question then becomes, and this is the interesting area of research, is what's the most important causative pathway here? Is it obesity leading to metabolic disease? Or is it all these other things leading to metabolic disease that also just happen to lead to obesity? I don't know for certain what the answer is here. The science is not 100% on this. As I said, this is an area of active research, and we'll come up against a lot of those in nutrition. Now, one important factor that is sometimes left out of discussions about the scientific method is the peer review process. As I said, if you have a set of conclusions, a set of results that you believe that are very good, you need to get those results out there. You need to publish those results, and you need to give other scientists, other experts, people who should know, a chance to go over your data, go over your experiments with a fine-toothed comb, and really determine, did your experiments test what you thought they tested? Do your results show what you think they show? The reason why peer review is so important is because everyone has a bias. This is just a fact of life. Everyone has their worldview, their outlook, that influences the questions they ask and influences what they see. It's a fact of life. Scientists understand this. And the awesome thing about science is that the scientific process tries its best to not let individual bias lead us down the wrong path. Because one person with their bias believes something is true, science does its best to make sure that if it's not true, it doesn't make it into the general understanding of how the world works. So peer review is a critical part of this. Basically, are your data, is your analysis, is it good enough to stand up to scrutiny by people who know as much about the subject as you do? Peer review by people that don't know anything about the subject isn't going to be valuable. Well, not as valuable. But peer review by people that are experts in the field, they'll find the holes in your research. For example, I say the sky is green today. I am absolutely, utterly, completely, 100% convinced that the sky is green. I look at the sky, I see green. I'm going to go to the website, I'm going to start a blog, and I'm going to share with everyone this most amazing discovery that the sky is green. Problem? That thought process that's going on in your head right now, the sky is not green. What? Is she crazy? That's exactly what the peer review process is. That is you peer reviewing my statement. I could start a blog devoted to the sky being green, or devoted to my going to bed sun-rising correlation. But if I had to subject that conclusion to peer review, the holes in my argument would be pointed out pretty darn quickly. So, in conclusion, never trust results that haven't been through peer review. You might just be looking at one person's individual biased crusade. Most academic journals are peer-reviewed. There are a number of peer-reviewed nutrition journals that are available through the SPSCC library on ProQuest, such as the Journal of Nutrition, and Metabolism, the European Journal of Nutrition. These are good resources for peer-reviewed, rigorous nutrition research. I want to talk just a little bit now about some of the types of studies that you'll encounter as you look through nutrition research. The first few types of studies are types of epidemiological studies. Epidemiology is the study of disease at the level of the population. So if you are doing epidemiological nutrition research, you're looking at a sample of a population and figuring out how nutrition affects their health. There are 3 types of epidemiological studies that can be done in nutrition: cross-sectional studies, case control studies, and cohort studies. Cross-sectional studies provide a snapshot of a population, simply what the population looks like here and now. If I were to draw an arrow, representing time, and here's the now, a cross-sectional study just looks at what's going on now. So if I took you all and asked every one of you what you had for breakfast and how tired you're feeling right now, that would be a cross-sectional study, just looking at the now. In a case control study, you're doing what's called a retrospective study. You look at the population now, and you divide them up into cases and controls. This is in the population that you're studying. The cases are individuals who are sick. If you're studying cardiovascular disease, individuals who have cardiovascular disease. If you're looking at fatigue, individuals who are currently feeling tired. And then your controls would be your healthy individuals, those who aren't feeling tired, those who don't have cardiovascular disease. Once you've figured out who your cases and controls are, you ask them to look back into their history. That's why it's called a retrospective study, because they're looking backwards into history. And ask them about their nutritional habits. Ask them about their diet in the past. A cohort study is a prospective study. If I take my arrow of time, look at the now. In a cohort study, I take a bunch of people at the moment, that's my cohort, and I follow them up into the future to see who becomes sick and who doesn't. So since I'm taking people as they are now and following them into the future, it's called a prospective study. All of these types of studies can give important information as to how diet affects our health. The statistics are a little bit different between case control and cohort studies, but you can take a biological statistics course in the future if you find that interesting. When looking at nutrition research, you also see various forms of experimental studies. An experimental study is where you are actually changing a particular condition. The epidemiological studies we just discussed are observational. We're asking people to report on what they're already doing. In an experimental study, that's when you take people or animals or cells and you change their conditions. In nutrition research, you'll oftentimes see experimental studies that are done on animals, oftentimes mice, sometimes monkeys. So if you took a bunch of mice, fed them a specific diet, one group an experimental diet and one group a control diet, then see how long they can run on a mouse treadmill. The problem with animal studies is that they may not be applicable to humans, and you always have to keep that in mind. For example, vitamin C is an essential nutrient for humans, but it's not for rats. So if you were studying the importance of vitamin C in rats, you wouldn't learn anything about how important vitamin C is for humans. So animal studies have their place. They can be really important for testing hypotheses in the early stages. Like if we knew nothing about cardiovascular disease, we could generate a lot of hypotheses and test them more quickly and more cheaply in animals. But we still need to do the human research to figure out if what we're learning is going to actually be applicable to humans. You will also sometimes see in vitro studies. In vitro literally means in glass. It's Latin for in glass. And what in vitro studies means is you are studying cells in a culture dish. They may actually be human cells, but you're still studying isolated cells. So for example, if I took a bunch of muscle cells, gave them specific nutrients, and measured, say, how much energy they made. Again, this has its place. It's very useful for testing early hypotheses, again, cheaply and more quickly. However, it does have a limited scope. If you are studying just a single cell type in a culture dish, you really aren't mimicking the situation that you find in the human body, which is much, much more complex. Last, but certainly not least, you have human intervention studies. Human intervention studies are just like the mice, but with people. You take two groups, you feed one a specific breakfast every day, and you measure their fatigue. You feed the control group a specific control breakfast, maybe, say, a high-protein cereal versus a regular breakfast cereal. Human interventions really can get at what is going to be relevant, what is going to be medically relevant in humans. However, they are expensive. It is difficult to control. You can't inbreed humans the way you can inbreed animals in order to get genetically identical populations. But human intervention studies are really what needs to be done in order to tell if an intervention is going to have a positive effect on human health. One of the hallmarks and one of the things to look for in bad nutrition science is inappropriate extrapolation from animal studies or from in vitro cell studies. Just because really super high doses of a particular ingredient, a particular additive, gives cancer to lab mice, doesn't mean that the same additive at normal levels of consumption would have any effect on humans. Just because a particular molecule, say an antioxidant, really protects cells in a culture dish from damaging free radicals, doesn't mean that the same molecule or the same the same nutritional supplement is going to work in the complex human body. We've covered a lot of ground in this video. These recap questions listed here, you should be able to answer after studying the material in this video. Some of these questions may be identical to ones on your study guide, but some of them may be a little bit different. You should be able to answer all of the nutrition science related questions.